Thank you very much for the kind introduction. Good morning, everyone. So I am really happy to be here today, and I would like to start by thanking the organizers to have given me the opportunity to present to you all the work of our lab today. A pleasure to start a session and start discussing about biology and neuroscience. You'll see I focus this talk mainly on biology, as my training was in biology, and I actually switched to computer science during my PhD. So I think I wanted to highlight this also in this interdisciplinary atmosphere here, and I think interdisciplinary meetings like this one are super important because we need interdisciplinary interactions to leverage, to make science advance, as I guess you already heard from the previous talks, and I hope that this talk will also convince you that interdisciplinary interactions are essential in neuroscience too. So first of all, I'm not sure all of you are familiar with astrocytes and glial cells, so I wanted to start by talking to you about neuroscience in general. When we think about it, the first thing that comes to our mind is neurons and their networks. So here is an image from a recently published model of a whole hippocampus, so brain region. But in these models, for now, we only consider neural networks. But actually, neurons, they only consist of half of brain cells. And the other half, which we call glial cells, basically meaning everything that's not neurons in our nervous system, is also very important for brain function. But historically, neuroscience has been a bit neurocentric and focused on neurons and their electric signals. And I'm going to present to you briefly during this introduction the roles of one specific subtype of glial cells that are called astrocytes that have been described early on already by Cajal in the late 19th century. You can see some of the images here. And basically, they account for 20 to 40% of glial cells, so it means 10 to 20% of all of the cells in our central nervous system. And what's really interesting is that they tile the whole brain, kind of occupying whatever space is remaining in the extracellular space. And one individual astrocyte is able to contact 100,000 neurons simultaneously, and that is in the mouse brain. But astrocytes in the human brain, they're 10 times bigger than in the mouse brain. So it means that, hypothetically, we're not sure yet because we don't have connectivity maps for now in the brain. It means that, hypothetically, in the human brain, one single astrocyte could contact up to millions of cells, which is crazy in terms of computational power that these cells might have. And that's some of the things we're interested in the whole community of what we call computational glioscience. And then importantly, as you can see on the image on the left here, they also interact with blood vessels. At this interface, they basically cover more than 99% of the whole vasculature in the nervous system, and they contribute thus to the blood-brain barrier. And so here is a schematic showing all of the different functions that have been shown that astrocytes are involved in. It goes from metabolic activity to repair in case of brain injury, brain development, so basically they're pruning synapses and are very important in synaptogenesis in that way, and also are central elements of homeostasis in the brain. So they control concentration of different ions in the brain, and thus they are essential to maintain possibility of action potential, so electric signals to propagate within neurons. So many different roles, and what we've been focusing on mostly in our past work was their roles in information processing, and more recently we started to investigate what they're doing at their interface with the vasculature, and this is a simple schematic showing you what will be the main focuses of this talk. So basically here you can see two neurons that are communicating, so presynaptic and postsynaptic, and at their interface, the synapse, there's an astrocyte around, and this tripartite formation is what we call a tripartite synapse, which, so in the hippocampus, so brain region responsible for memory consolidation, it could be up to 60 to 70% of synapses that would be tripartite. So yeah, bear in mind that not all synapses are contacting astrocytes, but we will focus on those ones, and you can see here an astrocyte in very close vicinity of the synapse. It can be less than 20 nanometers away from the center of the synapse, so basically nearly touching the synapse there. And there they're communicating with the synapses, and I'll show you a bit more details later. And another, what we call here nanoscale subcellular functional units in astrocytes that we're interested in is what we call the perivascular astrocytic processes. So what we call processes can be a bit weird maybe for other fields. It's basically protrusions, so parts of the astrocyte geometry that are in contact with specific interactors. So here perivascular because they interact with blood vessels and neural cells in general, and perisynaptic that contact synapses, and they're referred to as PAPs for the first one and PVAP for the second one. I'll try and not contract the words too much during this presentation. Please forgive me in case I use the PAP and PVAP denomination. Yeah. And then how do we study astrocyte function? So they do not display electrical signals like neurons do. So we have to find other ways to measure their activity. And what has been shown is that each time astrocytes are active, and it's also measured in vivo in mice as presented in this image here, you can see some transient variations in calcium concentration in the astrocyte, which is what we call calcium signals. And this is how the vast majority of the community is quantifying astrocyte activity for now. And so here I recommend, if you want to see some dynamic movies of astrocyte activity, I would recommend you to go to these studies. And basically, in this study, what they showed is that when the mouse starts running, you can see a huge increase in calcium activity in astrocytes. And basically, they've been shown to be essential to many different brain functions at different spatial scales. For example, neurotransmission at the synaptic level. And how do they show that? If they inhibit calcium activity in the astrocyte, then you don't have, for example, long-term potentiation, so some sorts of memory consolidation at the cellular level. And then they've been shown to be essential to various processes at the whole brain level, like cognition, motor, or sensory processing. And one a bit dystopic, but also very informative study that has been done to show that is a study where they used chimeric mice. Basically, they transplanted human astrocytes within a mouse brain. And as I mentioned, astrocytes in the human brain are 10 times larger. And basically, these mice were performing better in all cognitive tasks they were asked to perform, which suggests that astrocytes play crucial roles in those brain functions. And there are many more recent papers that suggest that astrocytes are very, very important for all of these functions. And so why is it challenging to work on astrocytes? First of all, the morphology is very, very complex. You can see here a 3D reconstruction that was made using stacks of images from electron microscopy. As you can see here, so that's an astrocyte with a resolution of around 50 nanometer cube. And also they're nanoscopic, so it means that most of their protrusions here, most of these processes, cannot be seen using conventional light microscopy. So for example, with confocal microscopy, you would see something like that. So a soma, large branches, and then kind of a cloud, where you would not discern individual branchlets, protrusions of the astrocyte. So for long, studies of astrocyte activity have focused on those large structures that were visible with conventional microscopy. And so most of the signals that we were recording were the first two categories here. And so these signals in the large compartments, they're very slow compared to signals in neurons. So historically, we thought that astrocytes were too slow to be able to take part in synaptic transmission and dynamics like that. But thanks to advances in microscopy techniques, some new types of signals have been detected in this cloudy region, which are called microdomain calcium signals, which occur in these nanoscale protrusions, and they actually correspond to the vast majority of astrocyte activity. So 80% of the signals in astrocytes occur in structures that are very complex in structure and that cannot be resolved with conventional light microscopy. So it's highly challenging to understand, to characterize their dynamics at all of the different spatial scales. And for now, how these diverse signals are encoded, how they propagate, and how they are encoded. So basically, do these different types of signals emerge from different mechanisms? So basically, we don't know. There are many, many open questions in the field. And yes, and then does that mean that when you see all of these different spatial temporal properties, does that mean that they have different functional roles? That each type of signal would serve different purposes in terms of cell-to-cell communication? Well, that's also unclear, and that's what we refer to as the calcium code, and that we would like to crack in the community. And so our goal in the lab is to better understand whether there's a causal link between these two very complex aspects of astrocytes, meaning the nanoscale morphology and their activity at the subcellular level. And that's tough to do experimentally, so our lab has been focusing on developing computational tools to answer these questions. And I will show you a few examples of studies that we performed to try and answer these questions. The first one will be focusing on the interface with synapses, and the second on the interface with blood vessels. So here I will pass because I cannot show you a movie, but basically these are very cool results from Misa Arizono that are published now that have been published for years now. Misa Arizono, who is now a researcher at Kyoto University, and what they did was very elegant. They combined different microscopy techniques to resolve part of the challenge I mentioned before. Basically, they used confocal microscopy to measure calcium activity in the astrocyte, and then they changed lens so that they could use a STED microscope and have a better resolution of the shape of the astrocyte with a resolution of around 100 nanometers in XY, which is way better than the 250 nanometer resolution in XY from confocal microscopy. And then they superposed the two images, and they were able to distinguish the morphology of the astrocyte where the signals were measured. And basically what they found was very interesting. So first of all, this is just a STED microscopy image showing nanoscale astrocyte morphology. So this is in live tissue. This is from organotypic hippocampal culture. And what they saw is that astrocytes, they don't display cylindrical, very homogeneous morphology at the nanoscale. It's kind of an alternation between bulbous structures that they refer to as nodes, connected to each other with thinner structures referred to as shafts. And correlating, they measured calcium activity in these structures, and they saw that calcium activity most of the time was triggered in the bulbous structures, not in the shafts, and that most of the time they stayed localized within a single node. So that was very interesting to them, but they wanted to go further in this observation, and they wanted to try and see whether there was a causal link between the two. Could it be that just having a morphology alternating bulbous and thinner structures would be enough to constrain signal propagation and calcium activity in general. So we proposed to them to build a computational model to explore this question. So when you want to build a model, the first thing that you have to do is really understand the system that you want to simulate, and also which hypothesis that you can make with all of this prior knowledge. And as I mentioned, what we know is that most of these perisynaptic structures are nanoscopic, which means basically you could imagine, if it was a cylinder, you could imagine something like 1 micrometer long, and then in radius 50, 100 nanometers. So it's really small, which means that when you take into consideration basal concentration of calcium in the cell, which is around 100 nanomolars, you would have only 2 or 3 ions in each of these processes, which is really low, which also means that you have to take into account probabilities of interactions between your molecules and ions in your system. So reactions are highly stochastic in such small structures. Knowing all of that and the question that we want to ask, whether there is a causal link between shape and function, then it defines the modeling approach that we can use to simulate the model. And here, stochastic spatial modeling. So to build this model, we used the software called STEPS that was developed by the team where I did my postdoc in Japan. And basically, this tool is a Python package that allows you to simulate reactions. So you have a mechanistic model, and then you simulate reactions in different morphologies of interest. So first you have to describe a biochemical model. So I did not have time here to go into details on how we build such mechanistic models. So basically you need prior knowledge on which molecules are important in your system, which reactions take place, what are the kinetics of these reactions, and then when you know all this, you can start building your model. And we can discuss later in more details if you're interested. It's based on mass action law and Michaelis-Menten kinetics. Then you feed the tool with your 3D structure, which can be something as realistic as this thing that's a part of an astrocyte that's directly reconstructed from electron microscopy. And then you can perform simulations. So you do basically experiments, in silico experiments on your system, and you try and answer the questions that you're asking. And so first of all, we validated the model by comparing dynamics in the model with dynamics measured experimentally by our collaborator. And we calibrated the model until we had no statistical difference between all of the calcium peak amplitude, duration, and frequency between the experiments and the simulations. So back to our experimental observations, we saw that there was a correlation between astrocyte nanoscale shape and activity. Is there a causal link? So to answer this question, it means that we need to model this node shaft architecture. So what we decided to do is use the quantification of node shaft with ratios measured experimentally, and to build 3D reconstructions alternating between nodes and shafts with these different ratios observed experimentally. And then to calibrate the model and see whether it was enough to reproduce the dynamics of the experiments, we performed simulations that were mimicking experiments that are done experimentally that are called FRAP experiments. What is it? Basically, you bleach part of the fluorescing molecules in the system. So the bleached molecules are the ones in yellow here. So at first, you have a baseline of fluorescence. Then when you bleach the yellow ones, they do not fluoresce anymore. So you have a temporary decrease in fluorescence levels, and then, so for example, measured here in the central node, and then what you measure with these FRAP experiments is the time it takes to get back to a new baseline of fluorescence activity. And then, so basically the time for the molecules to diffuse and for the whole system to be homogeneous again. And basically, the slower, the longer the time to get to the baseline, the more compartmentalized the structure is. And what we saw was in line with experimental data, so showing that this very simplistic model of astrocyte shape at the nanoscale was a good representation of what was observed experimentally. And that it was enough. Basically, what you can see here is that yes. So basically, when you have a smaller, a thinner shaft here, you will have a higher compartmentalization, so a higher time to get back to recovery. So then we wanted to investigate how it would impact signaling. So if you imagine here a sphere and a cylinder connecting it, the thinner the cylinder, the smaller the exit to go out of the sphere. So as we saw in the FRAP experiments, it means that you have a higher level of compartmentalization and less diffusion from one node to the next one. So we were expecting that if you have thinner shafts, you would have a similar effect on calcium dynamics. So basically, D shaft, which is D0/3, so a thinner one, would lead to lower level of calcium activity. And we were completely wrong. We saw the opposite. So this was intriguing. So we started to wonder, okay, how come with such a low probability of the molecules to diffuse away from the sphere, we still see an increased activity in the nodes? So this is just a quantitative representation of what you can see here in the representative traces. And basically, this is what gave us the answer and a full understanding, maybe not full, but an understanding of what was happening here. The thinner the shaft, the higher the residency time of calcium within your sphere. And then how does it lead to calcium activity? Well, calcium signals in the model, they result from the binding of calcium to a receptor that will then open and lead to more calcium increases in the cell. If you trap calcium within a smaller subpart of your system, you really increase concentration locally, and thus increase the probability of calcium to interact with the receptor, and thus increase the probability of the signal to be amplified. And even though you have less diffusion from the node to the shafts, because you have such a higher concentration of calcium in the node, overall you will still have a higher chance of a few ions to diffuse away and for the signal to propagate and then to go to the next sphere where it will be amplified again, etc. So that was very interesting to us. For time constraints, I won't show you other results from this study, but we also showed how such a morphology could impact signal propagation robustness, and also how it was altered in case of swelling conditions which are observed when there's a hypoxia, for example. Then I wanted to show you a more recent study that we've done, which is also on perisynaptic astrocytic processes. And this time it's based on electron microscopy data. So I'm not showing you images here. You can just see here in green, it's a 3D reconstruction of part of an astrocyte, which is made, same here, from Z-stacks of electron microscopy images. So yes, here EM is for electron microscopy. And then we quantified the characteristics of the different perisynaptic elements in the astrocyte, which we could do because we also segmented all of the different synaptic elements that were part of the images. And then we used the simulation framework, computational modeling framework that I showed you before, so that we could simulate the model of calcium dynamics in the astrocyte, but this time in more complex shapes directly extracted from electron microscopy. And then we could perform different in silico experiments, notably testing how the different geometrical properties observed experimentally could impact local signaling, such as, for example, surface volume ratio or the distribution of the calcium stores within the cell. And these are some of the predictions from the model. So basically predicting how each individual geometrical feature can impact local signaling in the astrocyte subcellular compartments that are directly communicating with neurons. And we saw that, for example, the endoplasmic reticulum, which is one of the calcium stores inside cells, it varies drastically in structural properties from one subcellular process to the other. And these properties will drastically affect calcium signaling, aside from any kinetics variations or local concentration variability. And then all of the tools and 3D meshes and code that we produce, we share in open access. And we hope that they will be useful to the community. And basically these things, so first showing that there's calcium stores in the perisynaptic compartment is changing the way we see astrocytes as perisynaptic structures, because it was highly debated whether they were present or not. So now we know. And here I don't have that much time, so I will briefly tell you that, sorry, I thought it was 30, I forgot it was 25. Basically perivascular processes, which are the ones interacting with blood vessels. Florian Dupeuple, who is a PhD student in the lab, he modeled these interactions between neurons, astrocytes, and blood vessels. And we started to have some interesting results. He's currently wrapping up his paper. And basically it seems that the distribution of synapses next to the astrocyte will completely alter the potential of astrocytes to mediate neurovascular coupling. So having synapses next to this subcellular region is key for this to happen. And then we also performed similar simulations in collaboration with colleagues from McGill in Alzheimer's. You can see that basically the astrocyte structure is completely changed in Alzheimer's. So for example, at the subcellular level, you have stacks of membrane on top of each other, and then we use computational modeling to simulate the effect on diffusion. And that's what you can see here. Basically, you have hotspots of calcium concentration in this Alzheimer's ultrastructure. And it seems that it's forming diffusional traps that are hard for molecules to escape. And in the next steps, we will investigate how it impacts signaling and not just pure diffusion. So yes, to conclude, basically we can use computational studies. At least that's what we do in the lab to perform some experiments that would not be feasible experimentally, or to infer causal links. And it's nice because it helps better understand the system. And it's basically back and forth loops, so a highly interdisciplinary field where we keep exchanging with people from the acquisition physicists, microscopists, and data analysts. And then the perspectives, I will leave them for discussions. You can come and talk to me. And I would like to thank my team and all of our nice collaborators and funders, and you for your attention. If you're interested in the field, I can share with you this slide, which is full of references and schools to go further. And I'll stop with this one. We are currently recruiting in the lab. So if you are interested or know people who would be interested, please do not hesitate to reach out to me. Thank you.